Hello everyone, my name is Arutha Kulasinghe. I'm from the Frazer Institute at the University of Queensland. Today I'll be talking to you about a really exciting collaboration with Akoya Biosciences and Navinci, where we looked at a cohort of head and neck cancer patients that were treated with immune checkpoint inhibitor therapy, and we had the pre-IO specimens. And this was in a cohort of 32 patients where we were looking at both the expression of proteins and the tumor microenvironment, but also the proximity ligation assays to look at the connectivity and interactions of PD-L1 and PD-1 within those cells. And so we used a combined workflow with this ISPLA panel up front on the same slide, and we stained this. And what we visualize is when you have these two cells next to each other, you've got a rolling circle amplification that lights up, which highlights the interaction between PD-1 and PD-L1, and you can actually visualize that. So it's this additive information from the spatial context that we're getting. And so we applied that workflow on the tissue, and we were able to identify areas of activity within the tumor microenvironment, but then also follow that up with the PSP panel looking at the immune contexture of the tumor microenvironment. And so we optimized the tissue in head and neck tissue, so tonsil tissue, which is a great organ to look at because you have all the antibodies, you have all the antibody production zones, all the immune cell types within that. So what you see here is DAPI to show you where the cells are, but CD8, PD-1, PD-L1, and that's the interaction that you're visualizing in these B cell zones within the microenvironment. But you've also got CD3, CD20, pancytokeratin, and CD8 within the optimized tissue. And so once we were really happy with the optimization of the samples, we went on to looking at this cohort of 32 patients using this combined ISPLA and PSP assay. And so we could see this gallery here, and this is representative of what we could see. This is DAPI, CD8, and the PLA expression in the green, and we can also see the CD3, CD20 areas. So it's really lighting up these CD8s and CD20 cells within the microenvironment, and this is sort of a composite image of all those colors together. And so when we look at the digital sort of maps and zoom out of that, we can see that there, and then we generate what's called a Voronoi plot to look at the microenvironment and the cell assignments of these cell types based on this multiplex panel. And so when we go into spatial analysis of this, what we have in here, these are the patients where an individual row represents an individual patient. This is the patients that had complete response, the patients that had aggressive disease, partial response, and stable disease. So we had RECIST criteria endpoints for this cohort. And so we could see the individual activity of the B cells, the T cells, the CD8s, and for every cell type, we could get the PLA expression profiles at the macrophages, the tumor cells, and so on. We could see the B cell zones light up here, and these were the T cell PLA positive areas within the microenvironment. What was really interesting was we could see this interaction as an additive function of what we were looking at. So we're really excited by this. This is sort of showing you the methodology that this workflow works on tissues. And so we will do deeper spatial analysis of this to understand what's defining response and resistance, but also areas of activity within the tumor microenvironment.